I want to talk about what we're doing, but I was asked to give a little bit of an overview on sequencing in general, and especially as it applies to PRRS. So I use the term next generation sequencing as a bet with one of my co-authors. We've got to get past next generation because we're calling everything next generation sequencing and it doesn't make sense anymore. So next generation sequencing is parallel sequencing, second and third, and maybe fourth. In situ is fourth. Parallel sequencing where you're doing multiple things as opposed to Sanger where you're sequencing one thing at a time. And I'm going to subdivide these, and you can argue with me if you want, into short reads and long reads. And short reads are what we are most familiar with. Examples of short reads are Illumina, Ion Torrent. The longer reads are Pacific Biosciences has a smart sequencer, and the one I'm going to talk more in depth about is the Oxford Nanopore Minion. So some applications of parallel sequencing. We can think about whole genome sequencing. We can do that with virus, but we can do it with bacteria. In theory, you can do a whole human genome for under $1,000. That's in theory. But more frequently, what they're doing in mammalian species is exome sequencing, so sequencing just the exons rather than the whole genome. RNA-seq, which we've been doing for a long time, especially with Illumina. But in PRRS, we've been doing something that I would classify as targeted sequencing, where we amplify OR5 and then sequence that with Sanger sequencing. We can also do that with parallel sequencing. Besides just amplifying with PCR, we can enrich in other ways. One of the new approaches is with CRISPR-Cas9. You knock out the ability to cleave, and you use that to target. You can do bait and capture. So those are examples of targeted sequencing. And then metagenomics, a buzzword that people use quite frequently, the microbiome, analyzing that. The first one I have there is actually a form of targeted sequencing, 16S sequencing, where we're amplifying the 16S sequence and then sequencing that. What we're going to talk about here in a little bit is shotgun metagenomics. I mean, that sounds cool, shotgun metagenomics, right? So what we do is just take everything, we throw it on the sequencer, and we see what we get. So again, back to the short read technology. These are some examples. There are more than this, but by far the most commonly used is Illumina sequencing. Really high accuracy. It's about as cheap as you're going to get for sequencing. The cost is in the amount of time for runs, and that if you're doing a small amount of sequencing, you have to wait for other runs so that you can batch those. There are some other ones. 454 came on the scene a little before Illumina and was popular, but really was hampered by slow throughput. Ion Torrent has a place. It's being used in human clinical medicine because of the short turnaround time. And then Solid is used in some instances. It's used in sequencing lymphomas in dogs and things like that. So long read technology. These are classified as third or fourth generation sequencing. Rather than having to sequence through different nucleotides, we have all of the nucleotides available in the PAC biosystems, and they're just detected by different colors. And so it could be a little faster runtime. And here we're getting long reads, and by long reads I'm saying several thousand bases rather than Illumina where you're maxing out at 300 or 400 or 500. And so if you're trying to map things together or you're trying to get more specificity, longer is helpful. The other one I want to talk about is Oxford Nanopore, and that's a little different in that we aren't sequencing by amplification, but rather by a change in current as it goes through a pore. And there we're getting long reads, and when I say long reads, 900,000 bases are what we can get with that. So here's the thing, and I'm going to pull out of my pocket my prop and put it on the table if you want to pass it around. It looks like a stapler. I think they should make it red. So the concept here is you have a lipid bilayer with multiple pores in there. And there's a current going through that pore, and if something goes through the pore at the same time, it deflects or changes the current that goes through the pore. So here's an example of the cartoon that we make. In reality, that pore is very small and the DNA is very long. But what we're getting is this squiggle of changes in current. And you can use that current squiggle to be interpreted as nucleic acid sequence. They've used it on the space station. This is in Antarctica. They used it in the depths of the mines. We're working on ways to prep samples. And they've also got one that just came out 2 days ago that's in alpha. It's not even in beta, that plugs into your iPhone. You've got to be an Apple user, I guess. But anyway, there is the ability or the appeal that this potentially could be a stall side test at some point. We're definitely not there right now. And the appeal to me was that they essentially give you the sequencer. It costs $1,500, but they give you those materials. So sequencing historically has been really expensive up front. Here, you can get that, and a library prep is $100. And if you buy the flow cells in bulk, they're $500 a piece. What's unique is it's a reusable flow cell, so you can sequence as much as you want and then stop. You can barcode to multiplex, and you can do sequential sequencing. So once you have enough, you can add the next sample. And we get real-time results. By real-time, I mean I put something on, and in 10 minutes, I have sequence data. So it's happening, and you can stop or change what you're doing based on the results that you get. So long reads averages about 8,000 bases. That's because we shear the DNA in order to get a consistent amount of ligation. It reads at about 450 bases per second per pore. We can get about 30 gigabases of data per flow cell. As little as 10 minutes for library prep with a transposon prep, and the sequence data is available immediately. The challenge, as with much of bioinformatics, is moving the data and interpreting the data rather than getting the data. Some unique things we can do with this: we can sequence RNA directly rather than having to go through cDNA. And a collaborator in the UK has something called read until, where we can determine what's coming through, and if it's not what we want, it actually reverses polarity and kicks it out. So we are enriching for the things that we want. It also allows us to sequence to a certain depth and then stop sequencing. And I mentioned the Cas9 approach for enrichment. So to the experimental question that we wanted to go at. PRRS has been sequenced, or OR5 has been sequenced for decades. And so genotype is important. It's a diverse virus. And so we wanted to see if we could identify and genotype PRRS from, initially we were going to target oral fluids. Somebody encouraged us to back off and do serum. Serum has been easier, but we've gotten it out of oral fluids. And then could we get some collateral damage? Could we identify anything else in addition? Because with PCR, you have to know what you're testing. Here, you just throw it on and you get what you get. So I'll skip these in the interest of time. So one of the things to think about was read identity. And so we said that Illumina is the standard because the error rate is 0.1%. For the Minion, when we started in the beta program, it was about 80% accurate. When you've got a really long read, that's still enough to correctly identify, but we want to get that better. And so the read accuracy has come up. So this is 2 months old, and we've got a new version of the base caller now, and we're somewhere around 93, 94% on the single read. But really what we want to know is how accurate is the assembly whenever you put them together. And there we're getting closer to Illumina. We're getting 99.5, 99.7. So it's still not as good as Illumina, but it's close enough that it's useful. So our workflow for what we are doing is an hour for our RNA isolation. We make cDNA. Then we take a couple hours for ligation. That's come down to a half an hour actually here in the last couple weeks. We can run that sequencer for up to 48 hours. And then we get to the analysis part, which can be 10 minutes if you have a fairly simple plan. It can be 10 days, or as Tanya Leroy says, you don't know what it means, so it'll be a lifetime before you can actually analyze anything. Maybe she's right. So the software, for us, we would love to take those 2-3 million reads and blast them, because that's straightforward. What we're doing instead is using a Python-based script called Centrifuge, which is for shotgun metagenomics. You can make custom databases, and I can ID each of those 2 million reads in about 90 seconds on my laptop. So it allows us to go through those fairly quickly, and we made a custom database of 781 full genomes that were available in GenBank. And then we use Pavian for visualization if we need to show the data. So initially, Oxford Nanopore made you send all the data to them so they could analyze it and figure out what you were screwing up. But they've gotten tired of getting all the data. And so now we're using Pavian. So this is a ribbon plot. This is just the virus portion of the ribbon plot. And we can see that we can correctly identify 25 reads for PRRS. And then a lot of phages. So initial testing, I'd like to thank my technician, who is a mean woman, and decided that we were going to blind me and then test PCV1 2A rather than give me something straightforward. But here the long read technology helped us, because I wasn't getting short reads that said PCV1 and short reads that said PCV2A. It correctly identified, and so I wouldn't have tried that hard, but she made me. So then we went to virus stock, what we had on hand, and we could correctly identify those. So now we need to get to actual biological specimens. So we had banked serum 5 days post-infection. We knew what we had infected them with, and so we could see whether we could correctly identify it. So experiment 1, we had 400,000 reads. The first PRRS positive read was in 5 minutes of starting the sequencer. We had 32 reads, 24 of which were unique to that particular virus. Second experiment, similar. We had 19 reads to PRRS, and 5 were unique to the virus that was used to infect. The third experiment, we had more unique reads to PRRS, but they were all over the place. So again, my technician, she chose to use a virus that we didn't have in our 781 genomes in our database. And so, in truth, what we ended up getting with all of these unique reads was, so here it was NADC 20, and we got 129, SDU 73, 163. So we were getting all of the viruses that were close, because it didn't have that actual virus in the database. So percent identity from just OR5 for NADC 20 is closest to this. We got unique reads that hit to there, but we didn't get to our other 2 viruses. So then I wanted to see whether our centrifuge approach actually allowed us to correctly identify whenever we had mixed samples. And so I mixed samples and ran those through centrifuge as well, and we got the appropriate number of unique hits, and they were the right reads. So I feel more confident. It still is a little bit of black magic, and so I think it needs to be tested more. But collateral damage. So on one of those samples, we also got porcine parvovirus 6, and a whole lot of those reads. So I asked when that serum was collected, and it was collected in 2013. Porcine parvovirus was first published in late fall of 2014, and then Ben House went back and found it in 2013. So this is an example of if we had been doing this kind of thing, we may have come across that particular virus much earlier than we did. So conclusions about this: identifying and genotyping PRRS is possible. Currently, we need to find a better way to enrich so that we can get a higher throughput. We've started barcoding samples so we can run multiple samples in the same library. An instance of this would be to, if we were going to sequence OR5, to take 96 OR5 amplicons, barcode them, combine them, and then make one library and run them together. Our analysis pipeline, the reason I say this isn't mobile yet, you can carry that thing around, but it takes about 10 hours of base calling in order to get, for 1 hour, 10 hours of base calling for every 1 hour of reads. And so we are doing this on the cluster, so it gets done very quickly for us, but it isn't something that would allow it to be a mobile system just yet. And we need to expand our centrifuge database, and we have identified other organisms concurrently. Some other things that we're doing at Virginia Tech, we're doing bacterial genome closure. We can do that for under $200. It's about $50 in materials. Amplicon sequencing for longer reads, looking at vectors. This will also allow for methylation analysis. PacBio and Nanopore allows for methylation, which may be important for oncology studies, not necessarily important for PRRS. And then we can do direct RNA sequencing. Our cDNA is going to be chunks that are about 2,000 or 3,000 bases. And so when we do direct RNA, we could rip through the whole genome fairly easily if we could just enrich for that viral RNA. So we're looking at how to do that more effectively, and also the Cas9 approach to that as well.